conclude today's first session, we have Professor Dr. Ron Heeren. He is a professor at the Maastricht University and will be talking about imaging spatial omics. Take it away.
Thanks very much, Donna. So, ladies and gentlemen, it's my pleasure to talk to something very close to my heart and very much in line with what you guys have been listening to today, which is complexity.
And think about it to really get the pleasure about the complexity, the inconceivable nature of nature.
That's exactly the problem we're trying to face.
But you gotta stop and think.
Sorry, I don't want to repeat Richard Feynman again, but that's exactly the problem that we're facing today, and this problem of complexity. And the only thing we've been doing today in all of the lectures that you've been seeing is we're adding complexity. Complexity of time and complexity of space. We're going down into single cells where all of these aspects in omics, whether it's genomics all the way down to metabolomics, play hand in hand with an environment that is continuously changing every second of the day of your life. So how do we tackle that? Well, in my mind, one of the ways to tackle this is imaging mass spectrometry, and some of you will know what this is. It's using a mass spectrometer as a microscope. And you might ask yourself, why do you want to use imaging mass spectrometry if we think about the clinic? Because I'm situated in Maastricht. The institute that I'm running is a multimodal molecular imaging institute. Not an element of cross-omics, but I have to talk to Alain about that. But we're doing a lot of omics inside the hospital. And one of the only reasons that we're trying to do this is actually to improve patient care. And that is why the role of mass spec imaging in clinical diagnostics is something that I'll be highlighting to you today and show you how single-cell imaging mass spectrometry can actually play an important role in improving our diagnostics, in improving high-throughput tissue typing using digital pathology, which integrates all of these different levels of information, and ultimately resulting in personalized medicine. And when we do that, we really take it from the single molecule to the patient. And just to highlight this whole range from molecule to man, I've actually highlighted here in a few examples of our own work where all of these images that you see down below are actually images generated with a mass spectrometer. And ultimately, we're currently applying them inside the OR where we're doing mass spec-based diagnostics during a procedure. I want to point out one thing, because you've heard Yvonne talk about mass cytometry. And that's a great imaging mass spectrometry tool that targets specific proteins. And I want to make that distinction very clear, that on the one hand, we have very targeted approaches where we label our antibodies. And just yesterday, for those of you who are keeping up with the socials of companies, Bruker announced their Ambergen alliance, where they're doing essentially the same thing that Yvonne was doing, but now we can do it on a normal MALDI mass spectrometer, targeting 1 to 40 or 50 specific antibodies. And behind me you see a bunch of very targeted scientists with their approach, and those are actually my colleagues from the clinic in Maastricht. On the other side, we have the untargeted approach, and that's where I'm sort of from, because we simply try to discover as many molecules as possible, whether it are metabolites, lipids, peptides, or proteins, from one single cell in the context of a tissue. And that's where imaging mass spec comes in the sense that we use MALDI-based technologies to discover as many molecules as we can see from a single piece of tissue. Now, if you do that and you bring that in the clinic into the pathology world, then there are three elements that are important. On the one hand, of course, there's MSI. Then you have to ask the right question. And this could be about patient typing, could be therapy prediction, could be tumor grading. And the way we do that now, considering the complexity of the data that we're generating, is using machine learning approaches. So the combination of this large data set that you're generating with all the mass spectrometers, the machine learning, and the spatial context of imaging mass spectrometry, that's really what brings forward diagnostics towards personalized medicine. Now, 5 years ago, when I was talking about these, we were talking about 50 microns, and we were, yay, we're happy. You know, we're seeing 50 microns, and we're doing a per pixel classification. But 50 microns, that's already, 25 cells. So at that point in time, well, maybe in 10 years we'll be down to subcellular levels. Well, I can tell you we're there already, and I'll show you some examples of how we went there. And that's, of course, all to make sure that we improve our clinical diagnostics. And this is just one example where we compare normal H&E-based pathology, which uses essentially an H&E stain, and look at the morphology or the morphometry of the cells in the tissue to classify, in this case, a specific gastric cancer. Now, if we augment that morphological information with molecular information, we find out that our diagnostics becomes 20% more precise. So in other words, by adding information, we're making our diagnostics and our treatment decisions more precise. It's essentially providing our clinical colleagues with better quality information so they can make more nuanced and better informed decisions. Now, if we do this with our molecular microscopes, there's a whole lot of work going on into improving our technologies. And we can think about improving the molecular content, the spatial content, and also about the throughput. I'm not going to go into all of these, but I want to highlight one recent development out of the group of Klaus Dreiswert at the University of Münster in MALDI, which is actually MALDI-2. And MALDI-2 is essentially a two-laser approach. The first MALDI laser creates a desorption plume, and the second laser post-ionizes the matrix neutrals to create more protons, which then enhances the ionization of the molecules that you want to actually look at. It enhances it by 2 orders of magnitude. 2 orders of magnitude means that I can either look at the molecules that I already was seeing with much more sensitivity and push my limit of detection lower, or, and in imaging mass spectrometry, sensitivity and spatial resolution are coupled, I can make the pixels that I want to look at smaller. And right now, where we are with the new instruments that are provided by a number of the vendors, and with the guys from Bruker, we've actually got it down to between 1 and 3 micron spatial resolution. So very similar to what you see in mass cytometry. So this is an example here of a timsTOF Flex with the second MALDI-2 laser built in. Just to show you some results of that, on the left you see some of the lipid spectra that we get from a single pixel, MALDI-1 and MALDI-2 compared, and this is the 2 order of magnitude more peaks and higher peaks of certain lipids. Translating that into the image quality, on the right side you see an image of a human glioblastoma where we have single cell sensitivity with this approach. So the question then becomes is, how do we use that? Well, that's where the digital pathology side comes in, and our realization that if we can look at single cells, why do we even want to isolate them? And I saw many examples. Harmjan was lysing cells, and I've seen the cell in one taking out cardiac myocytes. But if we do that, we lose the context of the cells. What if we could do that with imaging mass spectrometry and look at this one single cell and do the metabolomics or the lipidomics, unfortunately the in-depth proteomics that you're seeing, so we still have a way to go, but in the context of the tissue. And that's what Yvonne Verkoulen was also saying, right? Where we look at these networks of communicating cells and the pathways of how they interact. Now, that's exactly what a lot of researchers in Maastricht and throughout the imaging mass spec community are working on, because of this realization that we need to understand the context of a cell. And I'm showing you here some snapshots, right? But there's also time, and somebody will ask the question, yeah, how do you get dynamics involved there? Well, there are ways. I'm not going to talk about that here, but you can do pulse chase with stable isotope labeling where you can actually look at the dynamics. Anyway, I wanted to show you how we do that and how we do the single cell in digital pathology. And normal pathology starts with an H&E stain. And I'm going to show you some examples of human intestinal tissue that was taken out of an ischemia reperfusion study, and I will come back to the details in a minute. And of course, we can zoom into these cells and we can start to do some morphometry. And that's typically what a pathologist does, right? He looks in detail, and he tries to identify every single cell, and is there something going wrong with the cell? So in this particular image, I think there are about 40 or 50,000 cells that we want to analyze. Now, if we have the optical image, we can already start to draw a line around every single cell and do a classification based on the morphometry. So the shape and the circularity of the cell and the nuclear density and the color intensities. And we find out that there are roughly 2 cell populations based on this machine learning algorithm that was trained by a trained pathologist. This H&E image was taken after we had done the imaging mass spec experiment, right? The imaging mass spec experiment shows us all these different, in this case, lipid and metabolite patterns. Now, with every single one of these patterns, we can identify an individual cell type, because there too, we now have single cell resolution. And if I now look at this, zoom in on this image, on the middle you see the morphometry image again, but on the right, you see that some cells that have been classified exactly the same on the basis of the morphology, all of a sudden have a different metabolic profile. So in other words, we're putting in a bigger differentiation in the cell types that we're seeing because we're adding this molecular information to the mix. And then of course we can project that information back to the normal cell types. So this is now the routine strategy that has become routine in our lab, and many of my colleagues use that. And one particular study that we've been engaged in, where we use, among others, this technology, is actually in collaboration with Maarten, who was sitting there on the 3rd row, where we actually combined imaging, the way we do it, but also the classical omics. And the way we combine that is in this study where we actually take intestinal material from patients, is actually cut out, and during a Whipple surgery, a vein is clamped off, which makes the tissue ischemic. Then after some time, the clamp is let go again, the blood reperfuses that particular tissue, and then the tissue is actually, the piece of intestine is taken out. That piece of intestine is always taken out during this Whipple surgery, so the patients don't suffer from it, ethically approved and all that. Then we did some imaging experiments on cryosections of that tissue, and we selected out certain cell types, and they were actually sent to Maarten for full-blown quantitative proteomics and phosphoproteomics. And this is our little workflow diagram, and I'm not going to go into detail a lot, but the top part is the imaging bit, the middle part is the proteomics bit that was done in Utrecht, and we tie that all together by getting what we call a spatial proteome. In our workflow, we always combine high throughput screening with high spatial resolution, but we also do high resolution analysis, because in this case we're looking at tryptic peptides on tissue. So these are tissues that have been digested, on tissue digestion, by spraying them with trypsin and then doing the tryptic peptide imaging. So what do we see? Well, first of all, we see beautiful cellular classification. And not only do we see beautiful cellular classification, but we also find the ischemia and the reperfusion injury back in the proteomic signatures that we see. And you can actually look at the behavior. It's a little bit similar to what Yvonne was showing based on her targeted proteomics, but this is now fully untargeted. We can translate that into pathways, and the beauty now is, with every pathway that we highlight, we can actually also show the image and the distribution of the associated proteins in that pathway. So really, now we're linking all these molecules that we are seeing untargeted with the pathways where they're active in. And that to me is really, really cool, specifically considering that we are actually doing this with single cell resolution. Right? So in the images, we can actually see these individual cells. And this is just an example where we've similarly identified one of the lamina propria cells and actually got a full metabolic pattern with about 5 pixels per cell. So it's not as good as the 80 pixels that you get with cytometry, mass cytometry, but the beauty is here we get all of the lipids and the metabolites and the tryptic peptides of this tissue anyway. So, I wanted to put one slide in just to show you how we can also use this into drug treatment and diagnosis, and that is with a newly developed protocol where we're actually looking at organoids, and we actually make what we call super organoids, where we pool a lot of them together, make sections, and then actually do our imaging mass spectrometry. And not only, and these are pancreatic ductal adenocarcinoma organoids, they're patient-derived organoids. And we can actually now figure out what happens with the metabolism after exposing them to drugs. And in one of the organoids behind me, you see this orange center. Well, these are hollow organoids. And in the freezing process, we actually captured all of the metabolites in this organoid in the one single image. If you want to read more about this protocol, this just recently appeared in literature. So, bless you. So what if we think about single cells? I've been talking about, we need to look at these cells in context of the tissue. What if we start with single cells? Would that offer us anything? So Eva Kuipers in my lab, she's actually a very talented young postdoc that now has recently become one of our PIs, and we were working together, among others, with Thomas Hankemeyer, and we're actually trying to think, well, can we actually do imaging mass spectrometry on cell cultures? So Eva started to culture a bunch of different cell types, and we found out, there are lots of different lipid profiles in this case, and we thought, what could we do with that? Well, let's see if we can build a library of 20 different breast cancer cell types, different genotypes. And what we did, we've imaged those, and we found out that if we look at the metabolic and lipid patterns, they beautifully classify. So you can build a model where, based on these single-cell lipid profiles, and you can average, 50 or 100 or 1,000 cells, whatever you want, and just to show you, we also see nuclear versus cytosolic lipids in this case. That resolution is there, and they classify very, very differently. So you need to look at what element of the cell are you building into your model. But if you do that, then they classify beautifully. And classification is great, but what does it bring you? Well, there's diagnostic value in there, because if you look on the screen behind me, you'll find out that some of these cells are PR positive, ER positive, or HER2 positive, or triple negative. So in other words, we can determine the hormone status directly from these cell types. Can we also do this in tissue? Then that becomes the next issue, right? So we have this database, we feed it into a model, and the model should be able to predict whether or not, if we find the cell during our image run, whether or not that cell is actually, what type of cell it is. And the answer to that is, with the right software, you can do that already while you're doing your imaging experiment. So while the imaging experiment is running, for every pixel that we analyze, we determine what cell type it is, what is the hormone status. In some cases, in this case, there's an outlier, so the model doesn't recognize that particular pattern, usually because that's a cell type that's not in the model yet. But the beauty is you can actually do this diagnosis very, very rapidly. And if we project that onto, for instance, human brain material, in this case, again, the glioma, we're now identifying, so we did something similar with neuronal cells that we're working up at the moment, and we can identify all of the different cell types, do the correlation analysis, do the neighbor analysis that Yvonne was showing as well, and actually try to understand how these cells communicate, how do they recruit the immune system, what metabolic signals are sent, on all these different levels of omics. So what's the next step? So this integration of imaging and omics, how could we do that differently? And there's this protocol called the spatial omics approach that you might have heard, and we're doing some of that where we're actually using now our mass spec-based images to guide our proteomics efforts. Yeah, so if you can cut out 1 cell or 10 cells or 1,000 cells from a piece of tissue with a laser capture dissection microscope, you can actually start to pool these different levels of information together, similar to what we did with Maarten, but that was then done on larger pieces of tissue. Important there is that you have A, the right workflow, and B, the right target. Because if we do normally laser capture microdissection, you will have these PEN membranes, these plastic membranes upon which you have your tissue, you cut them around and they drop off, or you shoot them off in an Eppendorf. But our imaging mass spec experiments are predominantly done on indium tin oxide coated glass slides. So we wanted to compare what are the amount of proteins that we see on the PEN membrane and what are the amount of proteins that we see on the ITO slide. And clearly, you see here behind me, it has an effect. We see much less proteins directly from the ITO slide than from the PEN membrane slide. So typically for frozen tissue, we use the PEN membrane. For the FFPE tissue, for frozen tissue, we use the ITO slides. And for the FFPE tissue, because that's preserved already, and we have to do lots of treatments anyway, we typically use the PEN membranes. But if you do that, you can actually start to identify all of the different cellular components. Typically, we see anywhere between 2,000 and 4,000 proteins from these experiments. Of course, it depends a little bit on how many cells you take. This was done some time ago, but with the new approaches on the timsTOF, we actually can go down to very short analysis time and reasonably large depth of coverage in the proteome. And again, make that into a pathway, so you actually see what type of pathways are activated. And the beauty here is, here we took a lipid image and used the lipid pattern to select an area and look at the proteome in that selected set of cells. And funny enough, we find all kinds of pathways that are related to lipid synthesis. So the choices that we make in what model do we pick also give us a little bit of bias in the answers that we're getting. The last bit I wanted to check on, because then I've ticked all my cross-omics boxes. So we had metabolomics, lipidomics, and proteomics. We haven't talked about glycomics yet. So I wanted to share with you one study, and then I'm going to finish. I have 10 seconds left. I'm going to finish by showing you how we used all of this in a warm autopsy tissue microarray from Johns Hopkins Medical School, where we've taken a per patient TMA, where every patient, the primary best tumor was sampled, all the secondary tumors were sampled, and all the normal tissue surrounding these metastases were sampled. We sprayed those TMAs with Pangase F from Rick Drake's group, and then did an imaging mass spec analysis. Combined that with our pathological annotation, with the resolution that we have, and then we actually found out that you can actually trace the glycan signaling pathway from normal tissue to the metastasis and to the different organs that it affects. So in other words, by combining these different enzymatic digestion strategies and combining the spatial and the omics information, we can now come up with better understanding, fundamental understanding of this case, metastatic pathways in breast cancer. I hope to have shown you how imaging, omics, pathways in the context of a tissue really provide much better insight in what is going on in a tumor biopsy taken directly from a patient. And that, I think, really helps us forward into making personalized medicine much more realistic by essentially doing a interdisciplinary single-cell omics approach integrated into our diagnostic pipeline. So, in other words, we shouldn't think about specific boundaries in omics anymore, but we should just apply everything we know to the problems that we face. So I'm going to stop here, acknowledge all of these people, the funding organizations that paid for all of this. I'm going to thank my group, a great bunch of very, very talented and motivated people that really enjoy, in this case, Château Neerkanne of Maastricht. Good life in Maastricht as well. We have lots of vacancies, so if you're looking, contact me. And I'm going to stop here by thanking you. Normally I'm saying, if you're interested, come to Maastricht for imaging, but I'm going to make this slightly a twink on this slide, because I'm not going to say come to Maastricht for imaging, but I'm going to tell you come to Maastricht for IMSC. Because in August this year, Albert, Manfred Wührer and myself are chairing the International Mass Spectrometry Conference 2022, where we're hosting about 1,000 mass spectrometers throughout the world, and we would love to see all of you there as well. And with that invitation, I'm going to stop and thank you for your attention. And take questions.
Thank you very much. There's one question in the back.
I'm blinded by the lights here, so I can't...
Yeah, Peter from Antoon here. Fascinating talk. So can you, in order to apply this in healthcare, say, you need a lot of standardization, your machine models need to be generalizable, and there seems to be quite early times for that. So what do you do to standardize these type of imaging technologies so that you can really compare across batches and across...
A perfect question, and there's actually a lot of work going into that. So for instance, to give you an example, on the tryptic peptide images that we take, we put on protein standards to measure the effectiveness of the digestion. We put in synthetic peptides to actually test how well our matrix coverage is. And we use actually that to normalize some of the data there to ensure that the instrument is in good working order and the data is good quality. So QA, QC measurements, and they're different for different imaging experiments, are being set up and standardized. More importantly is how well do you train? Because your algorithms, machine learning is great, but it's only as good as the training of the algorithm itself. So We try to do that across a number of different pathologists that are actually involved to study the same problem, and they come up with slightly different answers, and there's not always agreement between pathologists. So we try to test that by essentially doing tumor boards. We're not there yet, but a lot of work is being done in the standardization. And we have a group of about 10 people working on just that. So yeah, very important point.